Today I will talk about molecular communication and various experimental platforms at multiple scales. So first I will give a brief overview about molecular communication concept, and then I will show you some of the experimental platforms that other groups in our group are working on. So we are living in a smart and connected world where we try to connect every physical object to the internet through telecommunications. Sometimes we call it IoT, sometimes we call cyber physical systems, ubiquitous connectivity. So the aim is to make everything smarter and connected. So to be able to provide this connectivity to all physical objects, engineers are miniaturizing electronic devices which can communicate wirelessly. But there are some challenging environments for radio signals that electromagnetic signals, radio signals do not propagate very well. Examples for these at the macro scale would be underwater. For underwater communication, instead of EM, there are solutions such as acoustic communications or others. Similarly, in the underground, RF signals do not propagate very well through the earth. And also in the body area, it's very difficult to communicate with wearables and implants, especially implantable devices inside the body. At the micro scale, at the smaller scale, there are these man-made devices in the micro scale or nanoscale. Here we see examples from the DARPA Shrimp Challenge 2018, like very small micro robots. To be able to control these robots, we need to provide them some communication unit. But when you shrink your device size this much, your antennas are also going to be shrinking. Then your frequencies are going into the terahertz range. And terahertz is still an active area of research for communication. We don't know exactly how we will establish communications at the terahertz level. Also, besides the man-made devices, there are engineered cells that can also perform some computational tasks. So how we can interface with these engineered cells? So on the considering all these challenging environments, we turn into nature. So how in the nature the communication is happening? So for example, we talked about the body as a challenging environment. Our cells communicate with each other by exchanging molecules. So in the eukaryotic cells, proteins, ions, or hormones are exchanged. In the prokaryotic world, when we talk about bacteria, they use autoinducer molecules or quorum sensing molecules to communicate with each other to understand their population density. That's how they change their behavior. They become virulent, they become infectious. So this communication takes these standalone biological devices, so each of these cells can survive on their own. But when they communicate, they cooperate to perform more complex tasks that they cannot execute alone. Eukaryotic cells come together to form tissues. Prokaryotic cells organize among themselves to create bacterial populations, bacterial biofilms. So especially bacterial biofilms that form on medical devices or when the infection case, they are the ones that make you very sick, and they are the ones that are more resistant to antibiotic treatments. So by getting inspired by the cell-to-cell communication in the nature, the field of molecular communication was born. So how about we exchange information by exchanging molecules instead of exchanging electromagnetic signals? So now we will encode the information on the molecules. It might be through a bio device or electronic or a hybrid device. And then these molecules are going to diffuse in the environment. They can diffuse freely, or they can be propagated by a flow of air or medium. And they will reach the receiver side, where which will have chemical receptors to decode the information that was encoded into these molecules. So in order to study this new field, we need an information and communication theoretical point of view, because cell signaling has been studied in biology. But what we need to bring is the communication theoretical point of view, so that we can model this communication system and we can mimic it for some applications, which I'm going to be talking about a couple slides later. So this is basically a very interdisciplinary field spanning nanotechnology, electrical engineering, computer science, biology, and medicine. So how do we encode information on the molecules? So we encode it on the physical and chemical properties of the molecules. For example, we can use the quantity or the concentration of molecules. If you don't send any molecules, can you see my cursor?
Yes, we can.
So if you don't send any molecules, that's going to represent bit 0. If you send a fixed amount of molecules, that's going to represent bit 1. Or you can have multiple concentration levels, which will correspond to multiple symbols that you can send. Or you can encode the information on the type. So if you send one type of molecule, it will mean bit 0. If you send another type of molecule, it will mean bit 1. And you can increase the types of molecules that you will use to increase the number of symbols you can use. Or you can encode it on the timing of the molecules. If you send it at the beginning of the time slot or at the end of the time slot, which we call pulse position modulation. So that's also another way of encoding the information. And it's also possible to mix and match these. Okay, so we encoded the information on the molecules. Now, how they will be propagated from the transmitter side to the receiver side. So here we list some of the propagation mechanisms that are studied in molecular communication literature. At the very short range, within a single cell, we have these molecular motors carrying cargo on top of the filaments that are going everywhere within the cell. So this is at the very small scale how molecules are transported. Also, in the short range, we can talk about molecule diffusion at the synapses among your neurons. So these neurotransmitters are sent from the transmitting neuron to the receiving neuron in the synapse, and their concentration is going to change according to the action potential that was transmitted through this neuron. So it's going to transmit some information from the presynaptic neuron to the postsynaptic neuron. If we go up to the medium range, micrometers to centimeters, we can talk about chemotaxis. So in chemotaxis, the bacteria actively carries molecules, especially the DNA plasmids that we can engineer the bacteria with from one side to the other. So this is one form of an active transmission. And in the long range, we can talk about molecular advection and diffusion. And an example for this would be pheromones and pollens. For example, if a tree or if a plant is attacked by a bee or by an insect, they release some pheromones to alarm nearby trees or nearby plants to cover their leaves with anti-bug repellent molecules. So these pheromones propagate in the air with the advection coming from the wind in the air. So these are some propagation mechanisms. So in the molecular communication theory, we try to capture it mathematically using the Langevin stochastic differential equation. So we can have random walk. So there is no flow in the environment. The molecules just randomly go in different directions to diffuse. In the drifted random walk, there is the effect of flow. For example, if the molecules are propagating in the cardiovascular system, they are propagated by the blood flow. Or if we consider microfluidic systems, there is a flow in those microchannels that are pushing molecules from one side to the other. Or in the case of pheromones, it's going to be the flow of the air. And we can talk about active transport. The examples would be the molecular motors within the cells and bacterial chemotaxis, where bacteria actively transmits information from one side to the other. So what are the challenges of molecular communication? So classical communication engineering models are not directly applicable here because molecule diffusion propagates in random directions compared to our directional antennas. And there is a high delay because it takes longer time for molecules to diffuse one place to the other compared to the light speed of transmission of EM waves. So we need to revise the well-established concepts of communication theory, such as channel models, noise models, information capacity, and energy. On the other hand, we also need some new devices that can transmit and receive these molecular signals through chemical reactions. So let's see an example of molecular communication system. This was a system that I studied during my PhD at Georgia Tech. So we had a microfluidic systems where we had bacterial chambers, and we put engineered transmitter bacteria in one chamber and engineered receiver bacteria in the other. So when you feed this transmitter bacteria with sugar pulses representing a bit sequence, this transmitter bacteria produces this AHL, quorum sensing molecules. And the receiver is genetically engineered such that upon the reception of these molecules, it generates green fluorescent proteins. So by observing the fluorescence output, you can decode the signal that was received. So here you can see a video of a receiver chamber, how it is glowing up and down upon the reception of the molecules. Of course, this is a very sped up version of this video. Okay, so we defined the concept of molecular communication. So what might be the applications of this? So one application for molecular communication is to replace RF for replace RF for challenging environments. For example, in this work, the authors compared a Zigbee radio transmitter to a molecular transmitter that are transmitting through air ducts, which makes lots of sharp turns. So after a couple of turns, you lose line of sight with the radio transmitters, so you cannot receive a lot of information on the other side. However, with the molecular transmitter, since molecules can diffuse and cut those corners, you can receive so much more information with a molecular communication system in this challenging environment. Also, we can use it for unconventional computing platforms. We can use it for nanoscale networks or biological computers. So it is possible to engineer cells to perform like digital gates or digital circuits or analog circuits to do some multicellular computing. So if you try to engineer one cell to do all the computation, it's going to be very it's going to have a lot of burden on that cell. That's why we distribute these functions into multiple cells, and to transmit information from one cell to the other, like these are biological living cells, we use concentrate like molecules with different concentrations. This can be proteins, RNAs, etc. So within this biological computers, for example, it's possible to do a processor by engineering the DNA of the bacteria. So you can create mixed signal computation in living cells. And the information is going to be transmitted, like the signals that are going through the system are going to be in the molecular form. Similarly, it's possible to do memory with living cells. Here, we encode the information into the DNA of a bacteria into the bases, AGT, maybe you remember from your biology courses. So this is the original video, and each pixel of this video is encoded into the DNA of the bacteria with the bases, and at the end of the experiment, they kill the bacteria, they extract the DNA, and they decode the DNA and reconstruct the video. So it's possible to create biological computers out of cells, and molecular communication can be a way to provide communication functionality to these biological computers. Other applications areas is biomedical. So here we can talk about Internet of Bio-NanoThings. So right now there are a lot of wearable and implantable devices. These might be electrical devices such as brain implants, pacemakers, gastric stimulators, insulin pumps, or these can be engineered biological devices such as artificial organs, engineered immune cells, or engineered gut cells. So we need some way to make all of these devices connect to each other for a coherent application. So molecular communication can be a way to create this connectivity within the body where radio frequency signals propagate poorly. Also, we can use the existing communication infrastructure of the body to interconnect these devices. For example, there is a microbiome-gut-brain-axis where our nervous system and our hormones create a bidirectional communication within the body. So in this work, we try to study how we can connect devices into the molecular side or into the electrical side at the nervous system to use the existing communication structure of the body as an infrastructure to send artificial information to connect these devices. Also, we can use molecular communication concepts for infection detection. So here in this work, we are trying to eavesdrop to the natural communication of bacteria with our sensors and try to send it to the cyber domain for interpretation. So I will talk about this a little bit more in detail in the next slides. And then we can also use it as an extension for floating nanosensors in our bloodstream that are trying to understand, eavesdrop to the molecular communication of bacteria, creating the infection and report it to the outside of the body. So we talked about infection, we talked about bacteria. Why do we want to study the bacteria or infection caused by bacteria? Because bacteria as a unit cell is a standalone device. It can have sensing function, actuation function, communication function. It has a memory within its DNA. It can actuate with its flagellum. And it's easy to program these bacteria using synthetic biology. You can easily implement new functionalities within the bacteria. Also, it's easy to observe them through fluorescence, through motility, or through their growth. And they already exist in the body. There are billions of bacteria in our body, in our gut, in our skin. And in the event of their adverse population, they cause infection in different parts of the body. That's why we wanted to study this as a model system. Then how do we establish communication among bacteria? We can talk about molecules diffusing from one bacterium to another, which is called quorum sensing. There is a very nice TED talk on this, how bacteria talk. I highly suggest you to check that out if you are interested in how biology and communication comes together. Or we can use chemotaxis, where bacteria actively transports DNA from transmitter to the receiver. So there are a lot of literature on the bacteria-based communication, how we can generate different molecular communication signals with the bacteria, how we can create receivers out of bacteria. And the bacteria species, they don't live alone. There are multiple species that are living in the same environment. So they engage in social behavior. They compete with each other, they cooperate with each other. So we also studied that in our previous studies. And we tried to also quantify the information flow in a single cell. So building on top of this, the application that we wanted to apply this knowledge was the early detection of infections. So especially cancer patients, chemo patients, and cystic fibrosis patients, they are very vulnerable to infections, and it makes a huge difference to be able to detect these infections early to reduce the mortality and reduce the hospital costs. So this was a concept that we put down even before COVID. So after COVID, this became more relevant, I guess, although we proposed it for bacteria and COVID is a virus, but still I think some of the concepts might be applicable. So in the physical space, we are eavesdropping to the bacterial communication with nanosensors and sending it to a wearable hub, which can be in the form of a patch or a smartwatch, and they will use Bluetooth or other voice communication techniques to connect to a mobile device to send this physical sensing information to the cyberspace, where we can process it, secure it, and share it with emergency or clinics if we detect an infection, or we can create a clinical database out of this continuous monitoring of infection. So we modeled the infection here as a molecular communication problem. We said we are eavesdropping to bacterial communication. So how these bacterial communication molecules propagate in the tissue to reach the sensors. So the bacterial infection is our transmission process. The propagation channel here is the tissue, and the reception process is the reception by our sensors. We can also consider this as a MIMO channel, where you have multiple infection sites or where you have multiple sensors. How do you localize the infection with just receiving the quorum sensing molecules? So within the physical space also, we need to have some sensors. So these were the three alternatives that we proposed. We can either directly try to use electrochemical sensors to detect the quorum molecules, or we can have bacterial sensors which will take these quorum sensing molecules and convert them to some other molecules that can be more easily detected by electrochemical sensors. Or it is very easy to generate luminescence, fluorescence out of bacterial sensors. So they can, the sensor might have living bacterial cells in it, which will take quorum sensing molecules and emit light, fluorescence or luminescence for photodiodes to detect. Then after this electrochemical sensing, of course, we need to have some sensor interface to further send it down towards the internet. And in the cyberspace, we can use machine learning techniques with human in the loop to try to identify what are the cutoffs to say this person has infection or not, how in the time, like if we have the time series data, how we will try to process that to understand the progression of an infection so that we can detect it early, and how we should like securely and privately send this information to the cyberspace. That's also a challenge. So now I want to present some of the preliminary results that we had. We tried to target Pseudomonas aeruginosa infection. This bacteria is responsible for infections, for hospital-acquired infections and cystic fibrosis in burn patients and chemo patients. And it's very resistant to a large spectrum of antibiotics, and it can infect various organs such as lungs, urinary tract, kidney, and skin. And they have unique quorum sensing molecules that we can sense. And these quorum sensing molecules they can be easily found in bodily fluids such as sputum or blood in detectable concentrations. So we go back to our molecular communication abstraction of infection. We have the transmission process by quorum sensing molecule production by the infectious bacteria. And we have the propagation process, the diffusion through the tissue, and the reception process, the detection by biosensors. So we model the transmission process where we have the logistic bacterial growth. Infection is basically uncontrollable growth of bacteria. So normally we have some bacteria in our body, but they are kept in a balance through our immune system and through the other species of bacteria. But when we acquire infectious bacteria, they grow uncontrollably. So we have to take that into account. Then we model the quorum sensing molecule production, where we take into account the diffusion as well as production and degradation rate of these quorum sensing molecules. For the propagation, within the tissue there are cells, so these quorum sensing molecules are going to be following not a straight path but more like a wounded path around the cells. So for this we use the diffusion in porous medium equation here, where we have the concentration of the molecules depending on the diffusion as well as the production. And here the two important parameters are lambda and alpha. Lambda means the tortuosity and alpha means the volume fraction. Volume fraction is the ratio of volume of extracellular space to the volume of tissue, and tortuosity represents how cellular obstructions are hindering the diffusion, basically how long this path is compared to the direct path. So then we used console multiphysics tool to simulate using the transport of diluted species module to simulate the propagation of quorum sensing molecules from an infectious site to our BNTs or biosensors. So here we can see the result, where this side is the infection site, this side is the blood vessels, and here we assume that our sensor is somewhere around here. This is the distribution of the quorum sensing molecules. Then at the receiver side, for varying volume fraction and varying tortuosity, we try to plot the concentration that is reached. So here, when we reach 2 times 10 to minus 3 molecules for the quorum sensing molecule concentration, this basically corresponds to 10 to the 5 CFU of bacteria, meaning colony forming unit, like meaning the amount of bacteria which will correspond to this many quorum sensing molecule. We can reach that at around 2 hours. This is so much earlier than the regular mechanisms of detecting bacteria, where they take a culture from your throat, they grow it in the lab, which takes 16 to 24 hours. Then we moved one more step. So, okay, so we we modeled the quorum sensing molecules propagation up to the nanosensors, but how these nanosensors will report to the outside of the body. For that, we teamed up with TU Berlin in Germany and Lakeside Labs in Austria to come up with this concept where we have nanosensors that are floating through our bloodstream. They are constantly moving within the body, and they are equipped with nanosensors that can detect these quorum sensing molecules. Then when they detect it, their flag goes up, and when these nanosensors are passing through the heart, we put a gateway there. They transfer their information to this gateway. And at the gateway, we use machine learning to understand first whether there's an infection or not. Second, where does the infection was seen. So I'm going to talk about how we do that localization part as well. So here we have like two branches. In one branch, we use these console simulations to understand the when the nanosensors pass through an organ, will they go through that sensing region where they can sense these molecules or not, to create a detection probability for a given tissue. And on the other side, we will use a simulator that's going to map out the trajectories of these nanosensors within the body. So in the first part, we use the same example, like the same case as before. We try to plot the quorum sensing molecule concentration and try to understand how far these are distributed, so that like if our sensors that are passing through this region can detect them or not. Then in the second part, we use the Blood Voyager S, which is a nanonetwork simulation module capable of simulating the blood flow for all major vessels in the human circulatory system. It is modeled upon a simplified human circulatory system, so it maps out major arteries and veins and not a lot of capillaries. So it comprises of the 94 vessels and their respective blood flow rates. So you can, in the simulator, you can inject nanosensors from any vessels that you want, and you can observe their trajectories within the bloodstream with respect to time. So from that Blood Voyager simulations, we tried to find out how long it takes for one nanosensor to come to the heart, to start from the heart and go towards the body and come back to the heart. So like a round trip time. So that's important for us. Why? Because we use it to obtain traveling times per region. So how long it takes to travel through the feet or through the right arm, for example. And we can use that information to say, okay, this nanosensor reported some infection and it took them very long to get back to the heart. It means that they went maybe up to the feet or up to the knee. So then we use machine learning to do k-means clustering to cluster these traveling times. And we can see here that it is possible to differentiate between most of the body parts like thorax, hips, and knees. But for the middle section of the body, since like the traveling times will be close to each other, we have some problems there, like livers, kidneys, intestines. So they are kind of a bit merged. But other than that, we can at least see whether the infection was detected in the head, shoulders, etc. So we use those traveling times to create a decision tree model, so that when the next time we report a traveling time, we can say, okay, it comes from this part of the body or not. And we use this detection probability coming from the other branch to decide whether we have an infection or not and try to localize it. So our results here is shown for shoulders and upper arms. So here we plot the probability of successful detection with respect to number of nanosensors. So we see that when we increase the number of nanosensors, of course, the probability of successful detection is increasing. Also, among the graphs within the upper arms, what is changing between these three graphs is the number of threshold nanosensors we want reported, because one nanosensor can falsely report, but if we have 5, then we will have a more accurate information. So this is kind of an example to illustrate how we can use molecular communication concept and Internet of Bio-NanoThings concepts for biomedical applications. Now, I want to talk a little bit about the experimental platform. So all that we've done right now that I showed you is simulation. So how we can create experimental platforms for molecular communications to validate all the theoretical works, analytical works that we've been doing. So first, I want to talk about macro scale experimental platforms. So we talked about at the macro scale the communication among plants with pheromones, for example. In 2013, a group in Canada tried to build a experimental platform to represent the macro scale communication through the air. They have a spray that they can electronically control, and they have a fan to provide some flow to represent airflow, and they have the receiver on the other side that's gonna be detecting the molecules sent from the spray. They used vodka alcohol molecules. So there was an article about this setup in MIT Review saying sending messages with vodka. So here the idea was to create some environment at the macro scale with transmitters and receivers and one fan. So in my lab, I'm trying to build a more complex system as an extension of it, where we can have multiple transmitters, multiple receivers, and I'm gonna combine it with a structure compared to similar to a wind tunnel, so that we can adjust the velocity of the air that we blow or the direction of the air, so that we can target the receiver better. Or if we have multiple receivers, we can steer our molecules to go towards a predefined receiver. So the setup that I have in my lab right now consists of 16 fans that can be all individually controlled, and we have a transmitter as an electronically controlled spray, and we have the receiver on the other side. We use ethanol as the molecule. So you can see the fan setup here and the transmitter and the receiver. So the results that we got out of this setup, the preliminary results, are about the channel impulse response. So when we send a very short burst of molecules from the transmitter, this is what we have as a response curve at the receiver side. We compared that to the case where we did some experiments without the fans. You can see that with the fans, but first of all, both of these curves follow an inverse Gaussian curve, which we expect from the diffusion equations. Then we can see that both of these curves have long tails. This actually shows us that there is a possibility of severe inter-symbol interference. So if I want to send another pulse right at this time, they might be interfering with each other. And also, in the case of fans, we can see that the molecules are reaching faster than the case without fans. So this can increase the amount of info we can send in a given time. And also we can see that without fans, there is so much noise on the graph compared to the fans. So in the next steps, we want to extend this where we can have multiple receivers, and we can add the noise that's going to be in the natural air environment, not just like straight airflow, but with the noise and with multiple directions. So this was for the macro scale case. So we talked about bacteria, cells, etc. at the micro scale. How can we create experimental platforms for micro scale? So at the beginning of the talk, I'll talk to you a little bit about this microfluidic setup. So where we had engineered transmitter and receiver bacteria within the microfluidic channels. So there were a couple other works on microfluidic molecular communication. So this group from Boğaziçi University, Turkey, so they created this microfluidic channel with 3 transmitters and 3 receivers, and they have syringe pumps that they are pumping colored liquids through these transmitters, where they encode the information on the color. So at the receiver side, the fluid that is flown within this channel are accumulated into these reservoirs, and they have a camera that captures the images of these reservoirs, and according to the color, according to the amount of yellow basically in these reservoirs, they decide what was the information sent. Another microfluidic experimental platform, they used saline water, like salt water. They pump the salt water from the inlet and regular water in from the other inlet, and they mix it within the channel, and they at the other end in the reservoir they have a salinity based sensor, and according to that sensor, when they change the salinity of the input, they can detect the salinity change at the output and understand the message. So these were the experimental platforms in the literature. The platform that we built in our lab based on microfluidic channels looks something like this. So we have 3 inlets where we push colored fluids like red, green, blue, and we have a mixing channel, an observation chamber, and an outlet where we collect the waste fluid. So we try to encode the information into the combination of colors. So if we give like 0 green, 0 red, 0 green, 0 blue, so this symbol will be transparent. If we have only blue, only green, only cyan, these are going to be the bits that they correspond to. Or when we mix red and blue, we will observe purple and we will say, okay, the incoming bit stream is 101. So then what we try to do is to send the ASCII code for MSU. So this is the photo of the system setup. We have the syringe pumps that are pumping into this microfluidic channel, and the syringe pump that's taking out the waste. And we have a fluorescence microscope that can detect these color changes. And this is a close-up of the fabricated device. So with respect to different flow rates that we push the liquid from the inlet side, these are the results that we observe at the observation chamber. You can see that we can see the similar color pattern at the output. And according to this pattern, we can decode what was the information sent. So why did we change the flow rate? So in communications, our goal is to send as many bits as possible in a given amount of time. So we want to send it as faster as possible. So that's why we try to experiment with different flow rates in order to increase the flow rate, but still whether we can observe we can detect the incoming bit stream or not. So we are considering on extending this platform where we will send more bit sequences and try to find the limits of our systems, try to determine the data rate, also bit error rates, and have some detection algorithms from these images that we take, how we can, right now we are just doing it visually, but how we can understand it automatically from the RGB values of each of these pixels. So now I want to change gears and I want to show you one more example for applications of molecular communication and biological computers, let's say. So these are, I'm going to talk about 2 projects and I'm going to end there. So one of the projects that I'm currently involved in, which is funded from ARPA-E Hestia program, is strong and CO2 consuming living wood for buildings. So in this project, what we do is we take a wood scaffold and we infiltrate fungi and bacteria into these wood pores so that we can functionalize this bacteria and fungi to change the properties of natural wood, to enhance the natural wood. So once you push fungi and bacteria within these pores, they are going to grow and fill these pores. They are going to fill it with biomass. This biomass can be cellulose, can be biominerals, biopolymers. So that's going to strengthen the wood. And also, since these bacteria are living, when there is a, for example, cut in this wood, these bacteria can produce more cellulose, detecting that stress and filling that cut to self-heal the material. So we just started this project recently, so don't have any results yet, but I think it's a very interesting project. And so we want to make sure that we can control this bacteria and fungi remotely from outside of the wood. That's where our molecular communication concepts come into play. And another version of this project is recently funded through NSF, Engineering Living Lignocellulosic Matrix as Strong and Intelligent Building Materials. In this case, what we do is we take wood biomass and we separate cellulose and lignin, and we reconstruct it as a polymer bio-ink for 3D printing. And within this bio-ink, we add bacteria and fungi, and then 3D print different shapes within that. Again, the idea here is that we will make this bacteria and fungi programmable and remotely controllable, so that after you 3D print this material, it can be stimuli responsive, or you can change the material properties with sending signals to these bacteria after the construction of the material. So I tried to make a brief presentation about like the molecular communication concept, some applications, and some experimental platforms. So thank you very much for your attention, and I'll be happy to answer if you have any questions.